Hello everyone, my name is Shahan Danapatia and I'm going to present to you a paper called Monomeric CRP as a regulator of propertin. It's written by the authors stated below, and the presentation is by myself. So the main premise of this particular paper was that the activation of the complement system can negatively impact the epithelial cells of the renal system. What this means is that the complement system proteins can actually bind to the epithelial cells of the renal system and cause them to be damaged. There are proteins in the renal system, however, that do inhibit the activity of the complement system, and these proteins include the C-reactive protein, CRP. In this particular experiment, multiple forms of CRP, specifically monomeric and pentameric CRP, were tested to verify if this claim is true and whether or not the cells are protected. So the main reason that this particular interaction between the complement system and the renal system is a problem is that the complement system proteins can attach to the epithelial cells of the renal system on the surface and cause the cells to lyse and leak out their essential proteins. This in turn causes the cells to die and renal failure to occur. The main symptom of renal failure, proteinuria, which is the excretion of proteins through the urine, is also caused by this. The other issue is that the complement system is still necessary to remove necrotic cells from the body and keep infections in self cells from spreading to each other. So the question presides on which mechanism actually wins out. So a little bit of background knowledge is necessary in order to understand this experiment as well as the results of the experiment. The complement system, specifically the alternative pathway of activation, and the unique renal interactions that occur between, and how the body does compensate for the fact that the renal system can be attacked. The complement system is composed of a variety of proteins that can be activated by the humoral or antibody-mediated immune response of the human immune system against pathogenic cells. The actual function of this portion of the immune system is to help the cellular part of the immune system by causing inflammation and marking particular antigen-exposing cells for phagocytosis. The other portion of this immune response is perforating necrotic cells with membrane attack complex. Membrane attack complex is composed of C9 proteins of the complement system and opens a hole that lyses a particular cell and spills out its contents. There are multiple activation pathways, which include the lectin pathway, the classical pathway, and the alternative pathway, which is stabilized by propertin. The alternative pathway is also what's being talked about in this particular paper. For the alternative pathway, on the right, it is figured at the very bottom. The C3 convertase is matched together with a propertin to stabilize it and turn it into C5 convertase on the surface of cells, thereby allowing it to form the C9 pore. Although it might be a different binding site on the surface of pathogenic cells, heparin sulfate on the surface of proximal tubular epithelial cells allows the C5 convertase complex to bind and activate using propertin. The complement system can be inhibited in specific parts of the body by particular proteins or other molecules. In the renal system, this is done by C-reactive protein, which comes in two types, monomeric and pentameric. The inhibition occurs at the alternative pathway specifically and at the binding of propertin to the complex. The actual tests done in this experiment were performed on both necrotic cells of the Jurkat cell line and proximal tubular epithelial cells of the HK2 cell line. This was done in part to show the complement system's interaction with its intended targets, which would be the necrotic cells, and our self cells, which would be considered by the HK2 cell line. The two tests that were specifically interesting were the ELISA and the flow cytometry they did, and these tests were performed to show the interactions between CRP and the complement system, specifically propertin. The first set of results shown were those of the ELISA test, which are in figure 3 to the right. This was performed using antibodies that were made inside rabbits against human CRP. Both types of CRP were measured for their optical density in the first part of this test and were normalized to be approximately 0 and equal. However, when you added diproperdin to the mix, it was noted that a significant increase in optical density happened to MCRP, but an insignificant increase happened to PCRP. This signifies that there is a binding of CRP, specifically MCRP, to properdin, whereas there is not to PCRP. The second half of the test used an increasing C3b concentration, C3b being the part of the complement system where properdin and CRP are supposed to bind. The addition of C3b and increase in it, along with the increasing trend in the optical density, shows that all three are interacting with each other. The results shown in figures 5 and 6 to the right are those of the flow cytometry tests that were performed. In figure 5, C5b9 deposition was examined, where C5b9 is the proceeding stage after the binding of properdin and CRP. It was noted that properdin alone, as well as properdin with PCRP, had approximately equal amounts of C5b9 deposition, showing that the complement system proceeded without any of the cells being inhibited. However, the C5b9 deposition for properdin with MCRP and properdin alone was very different. Properdin with MCRP was much lower than properdin alone, signifying that there was inhibition occurring. This is clarified in part C of figure 5. In figure 6, a similar type of flow cytometry test was performed, but this time on the Jurkat cells, or necrotic cells, that are the intended target of the complement system. Instead of recording the amount of C5b9 deposition on the surface of the cells like they did in figure 5, the scientists recorded the amount of properdin on the surface of the cells. This was done in order to examine whether or not the inhibition of the complement system occurs as a result of CRP binding directly to properdin, or CRP binding to the surface of the properdin during the properdin complex. It was noted that the amount of cells with properdin bound to the surface after being incubated in properdin alone was the exact same amount as those that were incubated in a mixture of properdin and PCRP, saying that PCRP did not bind to properdin and inhibit the binding of properdin to the surface of the cells whatsoever. However, the opposite was true for MCRP, where the amount of cells that were expressing properdin on the surface was decreased greatly, showing that inhibition occurs directly from MCRP binding to properdin. This also shows that it binds to both the necrotic cells and the PTECs in order to inhibit the activity of the complement system. So for a bit of thought-provoking discussion, I'll propose the question on why monomeric CRP can bind to and inhibit properdin, whereas pentameric CRP cannot. I also propose the question, why would the body also stop the binding of properdin to necrotic cells when they're supposed to be the cells that are removed from the body?